Right. Good evening, students. Hello. Good evening. Good evening. I hope you can hear me loud and clear. Right. So we'll continue our discussion. Um, People, please take the MS CAM paper. Ne? Take that CAM paper. We have to discuss this first. We'll quickly go through this. Overuse cause depletion in natural resources. Notation. We'll take that. Will overuse cause depletion of natural resources. I'll take that as a correct statement. Then production of disease-resistant varieties of plants and animals help to reduce loss of biodiversity. What do you say that, people? Production of disease-resistant varieties of plants and animals helps to reduce the loss of biodiversity. Varieties of plants and animals help to reduce the loss of biodiversity. We didn't discuss that, neither. Reduce to loss of biodiversity. No, we said that this is helping in to improve the crop production. Wrong statement. HIV is one of the leading cause of death. And no, we said cancer. Wrong statement. Then cancer are also serious and growing health problem worldwide. Cancer, yeah, that's correct, people. Right. Next one. Cancers are also serious growing health problem worldwide. Cancer. Correct, neither. We said people, the heart diseases and the cancers. Is it HIV? No, these are very close statements, right? Is it given for HIV? What is exactly given in the resource book, people? Check it there. And cancers, I think HIV, neither. Cancer is also, I think cancer is a leading cause of death, how it goes. And then say HIV is a serious growing health problem. So it's, I have taken it, right? Like that. Then next one, will DNA fingerprinting is used as solving some legal issues such as immigration dispute. Yeah, that's okay, correct statement. Sum of all chemical activities of an organism is anabolism. No, we said metabolism, right, people? Wrong statement. Then movement occur as a result of irritability and coordination. Yeah, that's correct. And gene control specific physiological, morphological, and behavioral characters of organisms. Yeah, okay. Both addition and cohesion are help to result surface tension. No, right, people? Surface tension only due to cohesion, right? Not addition, wrong. But then transport of water, we can see that both are going to be helpful. Phosphoglyceraldehyde is a derivative of uh, tetrose. Phosphoglyceraldehyde. Phosphoglyceraldehyde is a derivative of tetrose. No, phosphoglyceraldehyde, glyceraldehyde. Derivative tetrose name in triose. Wrong statement. It's triose, people. Wrong statement. And then people, galacturonic acid is a component of middle lamella in fungal cell wall. Wrong name, people. Number of carbon atoms in monosaccharide vary from 3 to 7. In number of carbon atoms in monosaccharide vary from 3 to 7. Correct, ne? 3 to 7, right, people? And then next one. Number of carbon atoms of monosaccharide varies from 3 to 7. Correct, ne? The triose to the heptoses in monosaccharides. Number of carbohydrates in monosaccharides vary from 3 to 7. Correct, ne? And then next one, galacturonic acid is compound of middle lamella of fungal cell wall. Wrong. Fungi don't have middle lamella, right? Wrong statement. Right? Next one. Next one, people. Next one. Next one. Amino acids may have only one more carboxyl group. Correct, ne, people? Because the R group can have that too. So 13 correct. And then people, tRNA is a linear molecule with anticodon. tRNA has anticodon, correct, right? We learned that. And then people, mRNA has a complex irregular structure. No, right? Not irregular, ne? What do you say, people? rRNA, irregular structure, right? Not mRNA is complex, irregular, wrong statement. Ne? And then people, next one, the smallest RNA type is mRNA. Smallest RNA type is mRNA, wrong. Ne? What do you say, people? Smallest RNA, tRNA molecule, wrong. And then next one, people, NAD plus act as a reducing agent. NAD plus can undergo reduction, but that means it can undergo reduction. So it's working like an oxidizing agent. Ne? Wrong statement. NADP plus act as an oxidizing agent in cell respiration. NADP plus act as an oxidizing agent. NADP plus act as an oxidizing agent in cellular respiration. No, this NADP thing, oxidizing agent, correct, but you know, people, this is NADP is involving in photosynthesis, right? NAD plus act as a reducing agent. Wrong, ne? Oxidizing, ne? I made a mistake here. Right? Not reducing agent, oxidizing agent. Let me correct that. So it says reducing, no, wrong. So this is acting as an oxidizing agent in cellular respiration. Wrong. Why, people? Cellular respiration is a wrong thing. FAD act as a coenzyme. Correct, no, people? FAD plus act as a coenzyme. Correct, ne? Correct statement. Coenzyme. We learned that coenzymes, ne? Coenzymes. Enzyme helpers, ne, people, correct. And then ATP is universal energy carrier, correct. ATP universal energy carrier. So we'll take these statements on this manner. The only movement of organism occur due to irritability and coordination. Only moved, what is that statement? What is that statement? Irritability, coordination. What's that statement? Movement of occur as a result of irritability and coordination. Movement occur. So what is the issue? Only movement of organism occur due to irritability and coordination. No? What is that? Movement of organisms can occur due to irritability and coordination. You know, irritability is sensitivity, ne? And then the yeah. Right. Right. So I think that's all. That's all for this part. And then we are moving people to the next one. Production of disease-resistant varieties of plant and animals helps in reduce loss of biodiversity. We didn't say that neither. We didn't say that the production of disease-resistant ones are helping in finding people, what do you call, sustainable food production. Right? I think yes. FAD plus plus. Yeah, it should be FAD. Yeah, I made a mistake. It has to be FAD. Yes, not FAD plus. We didn't say FAD plus. Yes, right. FAD, not FAD plus. It has to be FAD. We learn people NAD plus, but it's not FAD. FAD. Yes. Yeah, I made a mistake there. Right, people. We'll move now to the next one. We'll move now to the next task. We have to discuss. We will discuss the BioEve paper fourth one. Find BioEve fourth one, people. Find BioEve fourth one. 2026 BioEve fourth. Right, we'll go with the first part, first question. Can you mark first? Can you mark first 4 question answers? First 4 marker lamai. Can you mark first 4 answers? Hurry up, people, mark them first 4. Hurry up, people, hurry up, mark. Can you? Oh, wait. I think wait. I didn't share, ne? Wait, sorry. All right, now okay, ne? Given below are some characteristics of life. Which of the following of these features cannot be observed within one individual organism? What is the answer, people? What can you mark first 4 answers? Lamai, can you mark first 4 answers? Can you mark first 4? Can you? Can you mark first 4? Come on. Come on, people. Mark, hurry up. Lamai. Can you mark? Are you not going to mark it? Go to annotation and then mark. Go to annotation, select the pen and mark. Given below are some characteristic features associated with life. Which of these following features cannot be observed within one individual organism? Evolution is the answer, right? Evolution is the answer. Right? So I'm taking red color. You mark some other color, okay? My answer is second. Question number 2, people. Number of essential elements found in human body out of the elements of living matter, 25. Neither. Answer is third. Very good. The compound contain only carbon, hydrogen and oxygen, and nitrogen is going to be hemoglobin, not glycine. Why did you take glycine? Carbon, hydrogen, oxygen, nitrogen. The compound contain only carbon, hydrogen, oxygen, nitrogen. I think glycine better, ne? Glycine better. Yes. Wait. Glycine better. Why? Glycine is an amino acid. Glycine is only this, ne? Because hemoglobin has some COOH, right? And this is NH2, right? Hydrogen and hydrogen, ne? So glycine better, people, right? Hemoglobin has iron. Then sulfur present too, ne? Glycine does not have even sulfur. And then people, question number 4, nitrogen containing carbohydrate is, chitin good. Chitin good. 4th one is the answer. This is first, 3rd one first. What act as a transport medium because of having? What act as a transport medium because of having both cohesive and adhesive behavior? I think people 4th is better, ne? 4th is better for that question number 3. He repeats 2 times, ne? Okay, question number 3. My numbering issue is there. And next one, viewers, second question number 4. Which of the following statement is correct regarding protein? Protein contains, I think people first one is the only answer. Other statements wrong, neither. And the usually secondary polypeptide chain forms the 3-dimensional, no, that's the tertiary level. Secondary structure of protein form linear arrangement of sequence of amino acid, no, that's primary. Only covalent bond types are present in the particular 3-dimensional, no. That's the other hydrogen bonds, weak interactions are there. Quaternary structure is a spherical structure of the polypeptide chain. The quaternary structure is a spherical structure of one polypeptide chain. No, more than one. So we have the answer for that, people. Let me clear your drawings. Thank you. So I think the answers up to 4th clear. We'll mark now, people, 5 to 8. Are you going to mark 5 to 8, my dear students? Are you going to mark 5 to 8? 5 to 8, Lamai. 5, which of the following contain nitrogen and phosphorus as a element in carbon, hydrogen, and oxygen? Which of the following nitrogen and phosphorus? Why do you mark the wrong ones, right? In such a big hurry, people. What is that? Nitrogen and phosphorus. RNA and collagen, nitrogen and phosphorus you can't take for collagen neither. I'm taking second one as the answer for question number 5. Second one as the answer, people. Phosphorus and DNA, no? What did you mark wrong one? Right? Question number 6, select the correct statement regarding carbohydrates. Select the correct statement regarding carbohydrate. Please mark with some responsibility. Ne? You are covering whole my screen now. Right? Select the correct statement regarding carbohydrates. Hydrogen and oxygen ratio of carbohydrate is 2:1. Wrong, nay, people. Hydrogen must be 2. So 2:1, not 1:2. Genetic material of organism contain carbohydrate. That is correct, people. So I'm going with the second one, I think. All carbohydrates are macromolecules. No, monosaccharides are not. All carbohydrate possess glycosidic bonds. No, monosaccharides don't. All carbohydrates are water soluble. No, polysaccharides are not. I'm taking second one as the answer. So whoever take the second one, they are good. Then people, if a DNA molecule contains 10,000 nitrogenous bases, 32 guanine, 32% guanine means people, right? That is equal to cytosine, so that it's going to be 32% cytosine. Therefore, adenine plus thymine is going to be, what is that percentage, people? Here, 64, 64 what is that? 36%, 36%. 36% divided by 2 18%. 18% of 10,000 means, people, very good. Second one, come on. Don't do it. I can't see the board now. I can't see my screen, right? Question number 8, people, which of the following is not a function of monosaccharide? Not a function of monosaccharide. Acting as energy source. No, they can. Acting as component of nucleotides. No, they can. Nucleotides can. Act as carbon dioxide receptor. Not a function. No, they work. Ribulose. You're going to learn it later. Ribulose. Ribulose is working like a carbon dioxide receptor. It's an upcoming lesson. Contribute for synthesis of macromolecules. Correct. And then acting as a, I can't read this question now, the way you marked it. Fifth one, what is that? Acting as a storage compound. Wrong, people. Fifth one is my answer. So that's wrong. Your answer, acting as carbon dioxide receptor, correct. It worked like a carbon dioxide receptor. Isn't it mentioned, people, somewhere in the under monosaccharide, right? They can work like carbon dioxide receptor. Ribulose is an example. It's photosynthesis. It's going to be important. So the answer must be, people, it is a storage material. Monosaccharides don't act as storage materials. We didn't discuss that. So the answer must be 5. I'm going to clear your answers, my students. I'm going to clear your answers. Clear all drawings. Harida, clear all drawings. Now wait, don't mark anything. Right? Is this okay? Now don't mark anything. Wait. Right. Now moving to next one. So possibly doesn't have nitrogen, right? No, sometimes some possibilities they have when choline part is there. Up to 12, can you mark? Can you mark up to 12, Lamai? Are you going to mark up to 12? Or are you going to mark up to 12? Select the correct statement regarding nucleic acids. Correct statement regarding nucleic acids. Correct statement regarding nucleic acids. What is your answer? Correct one they ask, huh? My answer is fourth one. Good, people, right? What is that, people? Contain carbohydrate. That's correct. Ribose is the carbohydrate. 2 DNA molecules are complementary. No, wrong. This is 2 polynucleotide strands are complementary. That is correct. 2 DNA molecules are not complementary to each other, right? Not 2 DNA molecules. 2 polynucleotide strands are complementary to each other. All right. So So I'm taking fourth one as the answer for question number 9. Question number 10. Select the incorrect statement regarding light microscope. Incorrect statement regarding light microscope. What is your answer? Light microscope, second one. Maximum magnification, 1000 times correct. Maximum resolution power is 500 times greater as a resolution power of human eye. Maximum resolution power is 500 times as resolution power of human eye. Now let's calculate that human eye, right? Human eye resolution power. Right, let's work that out. Let me do that workout. Okay. Human eye. Human eye. Human eye. Wait. Human eye resolution power. Wait, people. Human eye resolution power 0.1 millimeter. Right. Then light microscope, compound light microscope resolution power is 0.2 micrometers. You know 1000 millimeters, 1000 micrometers equal 1 millimeter. So therefore, if you convert the 0.2 micrometer, millimeter into micrometer, you have to multiply this by 1000. Then you have 100 micrometers divided by 0.2 micrometer. That means 1000 divided by 2. 500 times. Right? So the second statement is a correct statement. Second statement is a correct statement. Maximum resolution power is 500 times as the resolution power of human eye. Maximum resolution power is 500 times as the resolution power of human eye is a correct statement. So first statement correct, second statement correct. Any cellular organelle cannot be observed through it. Any cellular organelle cannot be observed. Yeah, correct. Light microscope does not show you, right? So why won't second come? Consist of branch, long chain. Nucleic acids are not branch, people. No. Nucleic acids are not unbranched. All right. So question 3, statement 3, any cellular organelle cannot be observed. Yeah, correct. Living and non-living specimens can be observed. Living and non-living specimens can be observed, correct. Resolution power is limited to the wavelength of light. We know, people, resolution power has some connection with the wavelength of light. So we know by improving the wavelength, we can improve the resolution power. So all these statements are correct. I don't have a proper answer for this. I don't have a proper answer. I'm taking all for this. I'm taking all. So can't we see the nucleus? We can see nucleus through light microscope. My answer is all. Right? My answer is all. Okay. So we'll just skip the question now. Right? So rest of the clear, eh? How to calculate the resolution power? So how to calculate? No, we can't calculate resolution power. You know, human eye 0.1 millimeter, light microscope 0.2 micrometer. So you take the ratio, it's then 500 times. Right? So nucleus is a cellular organelle, correct. So you can see the nucleus to the light microscope, but you can't see ribosomes. You can't see endoplasmic reticulum. We'll be talking about this in the coming week class, all right? So the cell wall is cellular organelle. No, we don't take cell wall as a cellular organelle. Cellular organelles, usually people, membrane bounded structures, we take them as organelles. Otherwise, they are structures. Right, now I'm moving to question number 11. Storage components which are non-polymer in an organism. Triglyceride is a non-polymer. Triglyceride is a non-polymer. Cholesterol is a non-polymer. Sucrose is a non-polymer. Lactose is a non-polymer. So except casein, rest of the one. So I'm taking A, B, C, D as the answer. Question number 12. Question number 12, which statement is correct regarding elemental composition of living bodies? 96% of living matter consists of carbon, hydrogen, oxygen, nitrogen, correct. 4% of organic organisms mass consists of calcium, phosphorus, magnesium, wrong, people. Magnesium, they have not taken for that 4%. They have taken it as minute content. 4% of organisms mass consists of mainly calcium, phosphorus, sulfur. Okay. So cholesterol is not storage. So what? So they're asking storage. Storage components which are non-polymers in organisms. Then we can't take cholesterol. Triglyceride. Storage components which are non-polymer. Triglyceride, okay. Yes. Cholesterol, then we can't take. Sucrose, correct. Lactose, correct. Yes, that's the fault there. Right. We'll fix that. Cholesterol, then I have to remove. ACD should be the answer. We'll fix this. ACD. ACD. Right. ACD. Why? Cholesterol is not a storage one. Yeah, that's correct. We don't store cholesterol. Right. Triglyceride is storage. Sucrose, storage component, sucrose. Lactose is storage. So ACD, correct. Yeah, correct. Question number 12, people. Which statement is correct regarding elemental composition of living bodies? 96% of living materials consist carbon, hydrogen, oxygen, and nitrogen. Okay. Right. And second one, 4% of organic mass is containing 4%. I'm taking it as wrong. Be wrong. See, 96.3% of human body mass consider carbon, hydrogen, oxygen, nitrogen. Yeah, we'll take that as correct. Right. And then people, statement D. Isn't that correct? Statement D. Boron, copper, chromium are some of the elements present in human body. Correct, people? Correct. And then carbon, hydrogen, oxygen, nitrogen, sulfur, iron, chlorine are micro elements. These are trace elements. Sorry, minute elements. We can't take that as micro because it's wrong. We said that is present in minute amount. And then some carbon, hydrogen, oxygen are macro. So wrong. So can we take people A, B, and then A, C, D. A, C, D, right? A, C, D. A, C, and D as the answer for that. A, C, D. All right. So I'm going to erase your answers now. I'm going to erase your answers. Right. A, C, D. All right. Okay. Right. Now we are moving people to next part. Right. We are moving to next part. Structure is a part. Right. Structure is a part. All right. First one, people, state the organic compound. Don't write anything. No, don't write now. Please don't write. No, you can't write. You can't write now. If you write, all of you write, then we won't be able to read it, right? I'll just handle that. Let me handle it. Right? Don't write. State organic compounds. State organic compounds found abundantly on Earth. You can chat with me, okay? State organic compounds. People don't say oxygen. What is that organic compound? Cellulose. Cellulose is the organic compound most abundant one, right? Give two structural differences between monosaccharide and disaccharide. What do you write, people? Monosaccharide, one sugar. You have write in detail, huh? One sugar unit, and then the other one, two sugar units. Two sugar units. Two sugar units. So isn't it cellulose or carbohydrate? Why did you write carbohydrate? No, organic compound, you can say cellulose neither. Right? Why you want to say carbohydrate? Right? It's an organic compound, right? You can say cellulose. Cellulose is a carbohydrate. Right. Second one. Resource book is given as carbohydrate, is it? Shall we check that? I don't have the book with me right now. Is it given carbohydrate? Then we'll take. Right? If it is given in the resource book, then take carbohydrate. Huh? Carbohydrate. Carbohydrate. Then take. You are responsible for that. Huh? I can't check it. I don't have the book right now with me. Okay. Next one. Okay. Okay. Fine. 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 Then we'll go with carbohydrate. Okay. Fine. So I'll then take cellulose off carbohydrate. All right. Then next one. Right. We'll take carbohydrate. Right. So we'll take next one. Next question, people. So you have to give two structural difference between monosaccharide and disaccharide. What else you can say, people? A glycosidic bonds present. Glycosidic. No glycosidic bond and glycosidic bond present. Monosaccharide, no glycosidic. No glycosidic. No glycosidic. And then this is present. Glycosidic bond present. Right. And then anything else you have written? Anything else you have written? Anything else you have written? Right. Yeah, this is a result by disaccharides, a result by condensation, right? And then those things you can write. Right. Next one, name three main types of lipids found in organisms. Right. Name three main types of lipids found in organisms. What are those three lipids, people? Triglyceride, phospholipids, and steroids. So we'll take that. Fat, that's triglyceride. Phospholipids, phospholipids, phospholipids. And then what is the next one? Steroid. Steroid. Next one, some of the properties of water given below mention how those properties help to living organisms. Like cohesive behavior. We learn, people, cohesive behavior is going to be helpful in acting as a transport medium, right? Property, cohesive behavior, mention how this property help living organisms, right? Transport, to transport, to transport in plants, transport in plants. Okay, that's one. Second one, this can be useful, people, to form a water film, film for organisms to walk on water. I'll take it like that. Water, it can walk on the surface of water, right? Fine. Then people, ability to moderate temperature, how that can be helpful? Cool plant body, plant body, cool plant body, and prevent becoming too hot, prevent become too warm in sunlight. Okay. Next one, expansion upon freezing. How do you write that, people? Expansion upon freezing. This helps to survive organisms in aquatic bodies during winter, right? Helps to survive organisms, survival of organisms in water bodies in winter, because the whole water is not going to transform into solid, right? So this is going to be helpful for them. Right, moving people to next page. Next page, where is it? Here. Give three examples. Oh, next one. Give three examples. Give three examples for branch polysaccharides. Three examples of branch polysaccharide. What do you write? What do you write, people? Glycogen, eh? Glycogen. What else you wrote? Amylopectin. Amylopectin. What else you wrote? Hemicellulose. Hemicellulose. All right. Next one, state three main features of proteins. So can we write starch? Nani, it's not branch. Ne? Starch, if you write people, then amylose and amylopectin together, no? So it's not clear. Ne? So I think you better write amylopectin. Right? Next one, we will state three main features of proteins. I'm taking these three features made up of amino acid. Made up of amino acids. Right? Protein. First one. Second one, I'm taking composed of one or more polypeptide chains. Composed of one or more polypeptide chains. Third one, four levels of structure. Four levels of structures present. Four levels of structure present as, no, don't say water soluble, insoluble, right? That's a different story. Four levels of structure present, primary, secondary, tertiary, quaternary level. Ne? Primary, don't say amphoteric, that's amino acid. Now they ask in protein. If they ask amino acid, primary, secondary, tertiary, and quaternary. Right? Biopolymers and macromolecules. Good. And macromolecules. Some of you say that made up of 20 different amino acids. Okay, fine. 20 different amino acid. You can also say elemental composition: carbon, hydrogen, oxygen, nitrogen, and sulfur. Okay, right? Carbon, hydrogen, oxygen, nitrogen, sulfur. Fine. Right, moving to next question, people. What is meant by denaturation of proteins? What do you write, people? Denaturation of proteins. Hmm? What is denaturation of proteins? Loss of specific three-dimensional shape. Loss of specific three-dimensional shape. Three-dimensional shape due to alternation of weak chemical bonds. Weak chemical bonds and interactions. Weak chemical bonds and interactions. Take that as the answer. Give two functions of DNA. What are you going to write, people? Store and transmit genetic information. Stores and transmit genetic information. Stores and transmit genetic information. Right, from one generation to next. Second one, store genetic information for protein synthesis. Provide genetic information for, yeah, store or provide genetic information for protein synthesis. genetic information for protein synthesis. Fine. Now, store, transmit genetic information, and then store genetic information for proteins in that. Those are the 2 points given. We had to go with that, right? Control basic function of the cells and all. You can write that, right? But then try to give the 2 main functions which is mentioned, right? Then what is meant by base pair rule? What do you call base pair rule, people? Adenine bind with uracil. Sorry. Adenine bind with adenine pair with thymine. Adenine pair with thymine with by forming 2 hydrogen bonds. Then adenine pair with uracil. In RNA, adenine pair with uracil by forming 2 hydrogen bonds. In DNA, adenine pair with guanine by forming 3 hydrogen bond. Sorry. Adenine, guanine pair with cytosine. Guanine pair with cytosine. You write in words, huh? By forming 3 hydrogen bonds. Right? Then you can say adenine pair with thymine by forming 2 hydrogen bonds. Then you can say that people in RNA, right? Or you can just say adenine pair with cytosine. Sorry, adenine pair with uracil by forming 2 hydrogen bonds. So, what about purines bonding with specific pyrimidine? I don't think it's like specific enough. Right? What is meant by base pair rule? I think for the base pair rule, you must write the full information, how the hydrogen bonds and all included in that. Huh? Next one. Next one, we will categorize the RNA based on their function and mention their functions. All right. Now you write that messenger RNA. Messenger RNA. You mention 2 functions, mRNA, transmit genetic information from the nucleus, copy genetic information, copy genetic information from DNA. Second one, transmit to site of protein synthesis, or transmit to site of protein synthesis. To site of protein synthesis. Write it properly, yeah? Right? Next one. What is the next one? Will tRNA. Transfer RNA. Transfer RNA. What are you going to write, people? The transport amino acid. Transport amino acid to site of protein synthesis. Last one, people. RNA, ribosomal RNA. Ribosomal RNA. What are you going to write for ribosomal RNA? What is the given? Ribosomal RNA involved in formation of peptide bonds. Right? Form peptide bonds. Form peptide bonds. Oh, provide site for assembling of polypeptide chain. I think that's better. Provide site for assembling of polypeptide chain. Provide site for assembling of polypeptide chain. Can you write tRNA, mRNA, RNA? Okay, fine, you can write that. Name other nucleotides that are not found in nucleic acid. What do you write, people? ATP, NAD plus, NADP plus, FAD. So these are nucleotides and dinucleotides. What is meant by resolution power of a compound light microscope? What are you going to write, people? Resolution power, minimum distance between 2 points that can be distinguished. Minimum distance between 2 points that can be distinguished. Give steps in sequence for adjusting microscope to observe specimen through the compound light microscope. That you write in detail. This you write in detail. What are you going to write, people? I can't write it. You just check your answer, right? What are you going to write? Place the slide on the stage, right, of the microscope and observe under low power objective lens. Right? First one, place slide on stage and observe under low power. Number two, what are that, people? Number two, look through eyepiece lens and then bring the object in position. Look through eyepiece. Look through eyepiece. You write it properly. Yeah? It's given in your note. Look through eyepiece. Look through eyepiece lens and move slide and bring object into position for study. Third one, adjust the mirror. Third one, adjust mirror. Adjust mirror and condenser to give optimum illumination. Adjust mirror and condenser to give the optimum illumination. Fourth one, what are you going to write, people? Use coarse focus control. Use coarse focus control to get the image. Last one, people. Use fine focus control. Fine focus control to sharpen the image. When needed, last one, when needed, right? When needed, use medium power and high power objective lenses by revolving the nose. Revolve the nose and use medium power or high power if needed. Right? Those 6 points must be there in your answer. 6 points must be there in your answer. Right? 6 points should be there. And then, people, moving to the final part, this essay question, people, I failed to prepare the marking scheme for this, but we'll just discuss the question. And what it says, describe different microscopes, different properties of microscopes. Describe different properties of microscopes. Now, you know, microscopes are having several basic characters, right, people? What are those properties of microscopes? Now, we have light microscopes are there, electron microscopes present. Whatever the microscope type, we know, people, 2 main properties are there. What are those 2 properties? Resolution power present and then magnification present, right? Then describe different properties of microscope. Did you discuss electron microscope in the class, people? Did we finish that? Saturday, people? Sunday students, we finished that, ne? Sunday class, I think I finished. Saturday class, we did halfway, ne? Lamai? No, we didn't do, ne? Shall we leave this and I'll give a marking scheme later, right? I'll share it to the group, right? I couldn't finish the marking scheme preparation for the essay question. So describe different properties. You have to write the light microscope an electron microscope, a small description, and then people writing the resolution power and and magnification. Also, you must write the differences between light microscope and electron microscope. So I'll come to that later, and then we'll end up the today's discussion. Then thank you very much for your participation tonight, and I'll be meeting you people soon. Right, see you all. Bye bye.